everyone, and welcome to today's webinar, Flow Cytometry as you have never seen it before. I'm Christy Jewell of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. Now, we encourage you to participate today by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Jordi Petriz, Group Leader, Functional Cytomics Group, Germain Trias y Pujol Research Institute. And for a complete biography on Jordi, please click the biography window at the top of your screen. Jordi, welcome today. You may now begin your presentation.
Okay, thank you very much for this introduction. I'm Jordi Petriz, and I'm working at the Germain Trias y Pujol Research Institute, and I'm very happy to share the information I've collected recently using the CytPix, the Attune CytPix acoustic flow cytometer, and I would like to share with all of you the information during this last year in different topics I've covered in my institution located in Barcelona, Spain. This is a short introduction about the Attune CytPix flow cytometer because it's the fastest flow cytometer now on the market. The acoustic focusing allows it to run up to 10 times faster than conventional cytometers, and allows for an overall experimental design, such as detecting rare populations, one of the topics that I will cover today. It's a flexible and multiple configuration and upgradable. It has a modular design from 2 to 4 laser systems, and it can collect up to 14 colors on the flow cytometer. And it's available also with a violet 6-channel configuration. The most important thing is that it combines the classical flow cytometry approach using this acoustic principle, and also using imaging. And it uses brightfield imaging at high rate, at high velocity, and obtain up to 6,000 images per second, with this 0.3 micron size resolution per pixel and a range of 0.8 micrometer particle detection. It also allows to catch and combine different morphological parameters, as you will see, and it can be correlated in the conventional flow cytometry that we always obtain on the flow cytometer. This is locate the different events on the flow cytometer and then look at the picture that were, or the image that were that was collected. And also it can be combined with a technology or some tools, especially dedicated, used for size measurement, etc. The brightfield imaging is a conventional technique suitable for the observation of stained samples, and the specimen appears darker on a bright background. Produce a magnified image of the sample with sufficient image quality to see details in the image. And there is a very simple setup with very little preparation required, as you will see, especially for a red blood cell analysis. The illumination light is transmitted through the sample, and the contrast is generated by the absorption of light in dense areas of the specimen. And this is just to show you how the system can work with the acoustics on and the acoustics off. The acoustics on and the acoustics off allow to tightly attach the particle to the core the center of the core, and this allows to obtain a very large number of focused images. If not, if in the absence of these acoustics on, it will be very difficult to obtain a very nice images across all the sample collection. As you can see here, there is the acoustics off, and here is the acoustics on, allowing this perfect, almost perfect focusing during the sample acquisition. And this is an example of the cells at real time acquired on the flow cytometer at the same time that we are collecting the classical parameters of the scatter properties and fluorescence measurements. We are also collecting all this imaging, all this information, which allows to look at the true events on the flow cytometer. This is an important topic because I would like to show you here the presence of leukocytes, and in the region R1, the presence of apoptotic cells. We can, using these image acquisition options, visualize every event to a large number of events based on the number of cells or events that we are acquiring on the flow cytometer, and also the notification of data size with chosen options, and image in all events or within a focused gate. We can select different dots on the conventional plot of flow cytometry plot, and then look at the images that we have collected on the flow cytometer. And now we focused on the events of interest, and we are really interested in rare event detection, and the Attune CytPix system is a very good technology to confirm the true nature of the cells that we are identifying, as you will see. During the sample acquisition, we can obtain manually a tight focus because we can adjust the camera settings and look to the perfect focus on the flow cytometer. And this can be allowed at low but a high velocity or high sample rate on the Attune CytPix flow cytometer. By applying gating strategy using size parameters, we can also combine the classical flow cytometer measurements. And as you can see here, I would like to highlight the different parameters that we can collect, because we are, for the first time, adding morphology parameters to the classical measurement using fluorescence, such as the area, square microns, the perimeter, the circularity, the pseudo diameter, the major diameter, the minor diameter, the minor major ratio %, the eccentricity. And by combining all these parameters, such as disorders, the entropy, intensity, the average normalized intensity, the standard deviation of the normalized intensity, etc., and all these parameters that you can see here, we can use this method to discriminate healthy and pathology cells without staining, and also separate different populations that initially we are unable to separate using conventional flow cytometry. This is the classical scatter parameters, but also by using fluorescence. And then by combining all this power, we are able to obtain the best possible resolution on the cells that we are collecting on this kind of cytometer. This is how we can simply imaging leukocytes. And as you can see here, we can identify the lymphocytes, we can identify the monocytes and neutrophils and eosinophils here. And then by gating the different populations, obtain the different images that were collected on the flow cytometer. And these are the lymphocytes, these are the monocytes, these are the neutrophils, and these are the eosinophils. As you can see here, a picture, a very nice picture of a lymphocyte, a different picture for a monocyte. We can distinguish the cell membrane, the nuclei of the different cells. is just the neutrophil, and here is the eosinophil, as you can see here. Also, we can look at the marrow and obtain very nice pictures of the megakaryocytes. I'm sorry for the typo here. And look at these very nice cells present in the marrow. And also compare the different normal populations with here the lymphocytes, the monocytes, the neutrophils, and here plasma cells as an example of a pathology with an increased number of plasma cells, which is a multiple myeloma. And you can, in the absence of a staining, only looking at the brightfield imaging collected on the flow cytometer, we can compare the classical morphology obtained here with the lymphocytes and plasma cells, monocytes and neutrophils, and compare the different information that the system is providing. Also, and is really important because we are comparing the classical morphology obtained by microscopy analysis. This is the methods that the hematologists conventionally use to look at the different smears, just in the case of peripheral blood or marrow specimens, and we are always trying to apply minimal sample perturbation methods to better preserve the morphology of the cells that we are looking for. This is a paper published in Cytometry in 1998 by Ost and collaborators, using a method to avoid the red cell lysing of the erythrocytes. As you can see here, they combine the violet side scatter, and in this case with the red light scatter, to discriminate thrombocytes. This is platelets, erythrocytes, and here granulocytes, monocytes, and lymphocytes. This can easily be obtained on the Attune CytPix, and this is a experiment performed in my lab. In this case, using the violet side scatter, and here the blue side scatter, discriminating platelets, lymphocytes, monocytes, granulocytes, and here the erythrocytes. And this is a very simple method, just using a violet side scatter kit, and only by changing 3 filters on the optics on the flow cytometer, we are able to collect the violet side scatter and then combine this information with the blue side scatter to better discriminate the leukocytes and other cells, nucleated cells from non-nucleated cells, and platelets, with minimizing the process of sample preparation. This is an example on how we can use the analyzed whole blood analysis for the erythroblast population, which is more sensitive to the lysing solutions. Oops. And as you can see here, we can detect this population and these other cells here. We have mainly lymphocytes, monocytes, and neutrophils here, but based on the different differential absorption of the violet light, we can discriminate the presence of the erythroblast in this population. And then, by using this region and this region gate to the images that we have collected on the CytPix. And here, now we can better discriminate this population of erythroblast, mainly containing orthochromatic erythroblast, and as you can see here, with a very nice nuclei in these cells, and we can compare the other region to show the other more immature erythroblasts that are present in the marrow. Because we are analyzing these cells in the absence of erythrolytic lysing solutions, and we can obtain here coincident erythrocytes with the erythroblast population, but we can easily discriminate these cells on the CytPix and then look at the true nature of these cells. In this case, we can also discriminate here a abnormal erythrocyte, which is a chenocyte, demonstrating all this speculated cell membrane corresponding to a altered erythrocyte. In addition, we can look at the presence of rare cells and confirm the true nature of these events on the flow cytometer. And this is a summary of an experiment to discriminate the presence of dendritic cells. Here, the plasmacytoid dendritic cells. This is the conventional dendritic cells, and here basophils. And then we can gauge all these populations and look at the plasmacytoid dendritic cells. Also look at the conventional dendritic cells, and also to look at the presence of basophils. In this case, we are combining immunostaining of these cells to better discriminate the different populations, which is not possible in the absence of antibodies, of course. And then we are combining all this information to also discriminate, and as you can see here, by a different size of the pictures, and so I'm showing in this slide the presence of dendritic cells. It can be distinguished the different protrusions of the cell membrane, and as you can see here. But this was done using a lyse and no wash method, which is conventionally used in most of experiments, or in most of the immunophenotyping experiments using flow cytometry. But comparing these results with the minimal sample perturbation assays, this is a no lyse, no wash method, we can better discriminate the true morphology of the dendritic cells, as you can see here, and better compare this information with the conventional morphology obtained using optical microscopy, which is conventionally used by the hematologists to demonstrate the presence of potential leukemic cells, and we are using all this information to learn more in the field of basic clinic research. Also, we can monitor the potential interactions of different cells. And this is a case of a patient with lung cancer, and only a small fraction of the cells, the peripheral blood cells, were interacting with circulating tumor cells. In this case, it represents less than 1% of the cells that we are studying on the CytPix flow cytometer, and showing the interaction of the cytotoxic cells with tumor cells, and look at the nice interaction of this cytotoxic activity as it can be easily demonstrated also in the absence of a immunostaining. This is just to show you a whole picture of different stages of these cell-to-cell interactions, demonstrating the cytotoxic activity. Here is the cytotoxic cell interacting with the tumor cell. As you can see here in a different picture, we can monitor how after this cytotoxic activity and how this cell is killing the tumor cells, we can detect blebs on this cell, and also discriminate the presence of more blebs in a more active or killing process of the tumor cells. And this is a very really nice method to demonstrate how we can measure these cell-to-cell interactions and how to demonstrate a cytotoxic activity in peripheral blood, even looking at the presence of very rare events. Also, we can look at the presence of leukemic blasts. This is a case of a patient with acute myeloid leukemia, and we started to look at the morphology of these cells. And this is by combining also immunophenotyping at looking at the markers that were expressed by the blast cells. And here we can show the different cells and look at details on the intracellular pattern of this different bright field absorption, and look at the presence of bright field and high contrast internal structures and other dark dots which are related to different granules that can be observed in this kind of immature cells. Just to show you a magnified or amplified image, and this is a leukemic blast, and also we can discriminate the presence of the hour rods, and this is a hallmark for the acute myeloid leukemia, and easily confirm the true nature of these cells by morphology, in addition to the classical measurements that we are performing by immunophenotyping these populations. Also, we can confirm the presence of small leukemic cells, which is now here I combine it only one marker, just to show you how we can gate this population for the CD34 marker. This is a very large cluster of cells, and by combining this marker, the CD34 marker, with the forward scatter, look at the presence of small CD34 positive cells and large CD34 positive cells, showing how, I'm sorry, I don't know why I missed this information. This is the cells which are which they have the large forward scatter, and these are the cells that they have the low forward scatter, and confirm the presence of these cells, which is also relevant for the immunophenotyping of these cells, and how to classify and obtain better information of the true nature of these cells, and now confirming again the existence of our rods, and as you can see here in this large CD34 positive cell. In addition, we can distinguish healthy and non-healthy cells in human leukemia. In this case, we added a DNA marker, because this can be relevant for the altered DNA content of the leukemic cells, and then compare the normal or non-pathologic cells with a leukemia population. And as you can see here, there's a peripheral blood showing a different pattern of, it cannot be distinguished, the presence of lymphocytes, monocytes, and neutrophils. And we can see here the most of cells which are leukemic in this case. However, by using a very powerful morphology analysis on the CytPix, we can also look at the most important cells, and this is allowed by a system looking at the true processed cells, looking at only one cell per image, and also which is centered, and then by gating this population, these images, we are able to visualize the cells as possible on the CytPix. In addition, we can select only when we are measuring only one cell or only one particle, and also combine, in this case, with CD117, different fluorescence intensity, and by comparing the morphology of the cells, identify two main different populations. By using this sequential gating, at the end, we are able to separate the healthy cells, here the lymphocytes and neutrophils, and here the eosinophils, and separate this population from the other, which is mainly enriched in leukemic cells. By using this powerful gating, we can also compare the true DNA content of the non-leukemic population. This is the lymphocytes, monocytes, and neutrophils, healthy cells in this patient, and look at the DNA content of the leukemic population, and by overlaying these two DNA peaks, look at the extra DNA content of the leukemic cells, indicating that there is a genetic instability in comparison with the healthy cells. And also this can have an important prognostic value, even we are not using this cytometer for diagnostic procedures, because it's designed for research use only. But it can be very helpful for doing clinical research. And then let's look at the normal, population here of lymphocytes, monocytes, and neutrophils, and here the true leukemic population, indicating the different changes in morphology, and also to look at details on the brightfield imaging to confirm also the existence of our rods, as can be seen here. Also, by combining the conventional DNA staining, and then in our lab we are using DAPI for high-resolution DNA analysis, we can monitor the abnormal mitosis and multiple micronucleation in the myelodysplastic syndromes and others. We are also monitoring this in innate leukemia, but this is especially demonstrated in myelodysplastic syndrome. This is a so-called mitotic catastrophe phenomenon, and as you can see here, this is a conventional microscopy and analysis of these cells, with the advantage that we can collect a lot of events on the flow cytometer and look at the presence of very rare populations. And this is a problem, because this is generating the MDS is a heterogeneous group of disorders characterized by this myelodysplasia, increased apoptosis and senescence, and we can look at the presence of these cells on the CytPix. And and as you can see here, the different mitosis that we are collecting on the CytPix, and looking at these changes in the nuclei of these cells, indicating the important heterogeneity of these cells when compared with the classical conventional optical microscopy using these smears and the hematologists are using for classifying this MDS. And here a more detailed information by collecting these images, showing how we can detect these multinucleated cells, indicating this huge heterogeneity in these myelodysplastic syndromes. This is a slide that I've prepared for obtained from the same patient, indicating these changes in the morphology of the nuclei of the cells, showing a very nice comparison with the morphology that we are obtaining in the CytPix. Of course, the resolution of these images are done in a special clinical optical microscope, and it's able to provide more details in comparison that the details that we are obtaining on the CytPix. But at the end, we are obtaining the same information and confirm the true phenotype of these cells based on the conventional cytometry and the immunophenotype that we are also applying simultaneously to this imaging system. Also, we are looking at the presence of metastatic cells in melanoma, and this is the shape of these cells that can be observed using conventional microscopy analysis, and migratory melanoma metastatic cells obtain in peripheral blood this specific shape, which is like a oval shape, and can be easily distinguished when compared with lymphocytes, monocytes, and neutrophils, and easily discriminate the presence of these migratory melanoma metastatic cells in peripheral blood. Also, we can measure, as I was explaining in the beginning, the true nature of apoptotic bodies. And as you can see here, we can discriminate lymphocytes, monocytes, neutrophils, and then look at this region, apply and select only this population, and look to the true nature of apoptotic bodies as confirmed by this imaging system on the CytPix. And also look at the different subsets or the different changes in the chromatin condensation during apoptosis. We can see, as we can see here, the lymphocytes and monocytes and neutrophils. And look, this is a magnified section of this density plot showing a normal lymphocyte, apoptotic cells here in B, and also apoptotic cells here in C. And look at the differences in the chromatin compactation easily on the CytPix. And also monitor, as I was mentioning previously, to look at the cells at the 0G1 phase by combining DAPI staining at the cells in the S phase, and also look at the cells and the G2M population, and look at the double amount of DNA in the cells, even the post-mitotic cells, as you can see here. This is a very nice example because I would like to show you how we can use to study a patient that was diagnosed with anemia using the classical approach in my hospital, and we were able to perform a cell imaging analysis on the CytPix after diluting only 2 microliters of peripheral blood in Heinz the balanced cell solution in 1 ml to show the abnormal red blood cells. This is a case of poikilocytosis. And only by diluting these 2 microliters of blood and then by running the cells on the flow cytometer, we can easily confirm the abnormal presence of erythrocytes and look at the different shapes that can be detected by only in 1 minute of acquisition on the flow cytometer. And this is a platelet that is attached to this cell that can be also easily distinguished on the CytPix. As I was saying, we can perform, in this case, some measurements, such as the area, and then compare at the true area of this erythrocyte, and also take measurements from different cells, and compare this information with a different patient with a PNAH, an important alteration in the erythrocytes, showing this very large erythrocytes when compared with a healthy donor here. And as you can see here, there's an important increase in the area of the cells, just to confirm that we can also perform this kind of measurements using the morphology software on the CytPix. Also, looking at the multiple myeloma, we can also confirm the existence of Dutcher bodies. The Dutcher bodies were originally described in intranuclear inclusions in Waldenstrom macroglobulinemia, and they were later identified in other plasma cell disorders such as multiple myeloma. Then, and as you can see here, we were able to confirm the presence of Dutcher bodies on the Attune CytPix in this case in this region. We combined the DAPI analysis to discriminate lymphocytes, monocytes, and neutrophils, and then look at this rare population, which is less than 0.07% of cells, by combining a specific antibody to discriminate the plasma cells in multiple myeloma. And then look at the true nature of the cells, demonstrating how we can easily distinguish and confirm the true nature of the cells by the presence of Dutcher bodies in these cells. And here are a more detailed slide showing how we can visualize easily these Dutcher bodies on the CytPix, in demonstrating the presence of malignant CD138 positive cells. Even they are a very rare number of cells are demonstrated in this case. And importantly, when we can also perform an important measurement, because we are doing a very nice experiment using nanoparticle analysis. This was done using liposomes, and this was done. These liposomes were acquired on the CytPix, and they were stained with the NBD stain to study the cargo of these cells and compare the cargo accumulation. This is the dye staining of the single liposomes in comparison with aggregates. This is impossible with a conventional flow cytometer, because in these days these are very small particles, and we cannot apply the conventional singlet discrimination to separate single liposomes from aggregates. However, by applying this powerful software imaging analysis, we can discriminate the single liposomes, the images that we have collected showing only one liposome per image, and then also obtain and define a gate to have only one single particle. This is one single liposome on the Attune CytPix, and then compare singlets and all events, including singlets and aggregates. We can have here, we were demonstrating here the presence of only one single liposome in comparison with the aggregates here and all events, and look at the fluorescent profile of the NBD dye when we are looking to the singlets and when we are looking at all events, and then compare the different fluorescence profile of the singlets here and the aggregates here, demonstrating a different cargo for the aggregates and for the single liposomes on the CytPix NXT. And this is a different approach. I had the opportunity to collaborate with people working in northern Spain, and I was really interested and look at the microalgae on the Attune CytPix. Even though I'm not an expert in this kind of microalgae, and we were able to demonstrate the existence, based on the autofluorescence of this microalgae, identify different subpopulations based on the load of the different pigments that these microalgae can have, and by combining the yellow laser and the red laser, look at different subpopulations of these different microalgae, and by selecting the different clusters and the different populations observed here, we can confirm the presence of the different species that are contained in this suspension of microalgae, and identify the different subpopulations based on these different regions that can be obtained on the CytPix Attune CytPix flow cytometer. In summary, the CytPix flow cytometer is equipped with a high-speed brightfield camera that can capture images of individual cells as they pass through the flow cell, and this allows the users to visualize the morphology of their cells and to identify and exclude cell aggregates, debris, and other unwanted event. This is especially important and relevant for rare cell analysis because the CytPix flow cytometer software can automatically analyze these brightfield images to extract a variety of quantitative parameters that can be used to improve the accuracy of gating strategies precisely to identify the cells in low numbers. The CytPix flow cytometer can be used to collect brightfield images with and without fluorescent labels in basic and clinical research, and it is a versatile and powerful tool that can be used to improve the quality and quantity of data obtained from flow cytometry experiments. And is particularly well suited for the studies of cell morphology, again, the rare cell population, and label sensitive cells. Now, I would like to acknowledge the people involved in this work. Mike DeWard, which is one of the inventors of the acoustic flow cytometer, Pierre Leninan and Marjorie Joyner in France, Nicole Mattes, Daria Tayakina, Vincent Jones, and Sergio Ramon, one of the local engineers in Spain, the people in my institution at the Germain Biological Research Institute, Laura Garcia Rico, which is a postdoc, Rousset Salvia doing her PhD in lung cancer, the hematology people, Rebecca Jurado providing samples, Marta Vives and Lidia Almenara providing us the samples that we have used for liposome experiments, Teresa Moran leading the oncology team about the lung cancer experiments and other measurements such as in melanoma. Marco Antonio Fernandez, the core facility manager in my institution, which also is very friendly, always happy to help us, and Angeles Cid at the Universidad de Coruña in northern Spain by providing microbiology, as I presented in my last slide 9. I'm going to finish here, and I'm very happy to take any questions from the audience. Thank you very much for your attention.
Jordi, thank you so much for that informative presentation. We will now start the live Q&A portion of our webinar right now. So to our audience, if you have not submitted any questions yet, please do so now. Just type them into the Ask a Question box and click Send, and we will answer all of your questions that we have time for. Okay, well, we already have some great questions that have already come in. So Jordi, I'm gonna go ahead and start with this one. Where do the megakaryocytes show on the scattergram of the bone marrow? Oh, Jordi, you're muted. I'm not able to hear you. Let me double check here. Jordi, let's see. I see that you're unmuted, but did you mute from your computer possibly? Nice. Jordi, do me a favor. Refresh your browser real fast. Let's see if that fixes it. No? You can hear me though, right? Okay, perfect. I can't hear you. Let's refresh your browser real fast. Not yet. I can read your lips. Do a quick browser refresh. Thanks for your patience, everyone, while we get this reconnected. We have some great questions we want to get to, so give us just a moment. Not yet. Jordi, refresh your browser. Just click refresh and reconnect. And thank you, everyone. While you wait for just a minute, technology is great until it isn't. So just give us a minute. I can see that you have lots more questions coming in, and we'll take our time to get to those. So thank you so much for hanging in there with us. Give us just a minute while I reconnect. Yes, wonderful. Yeah. Excellent.
My microphone is not muted, and suddenly I lost. Of course, I can hear you, but I was trying to reply, but it was impossible. Sorry for this technical situation.
Not a problem. And as virtual attendees and producers, we all know that sometimes can happen.
I heard your first question.
Okay,
and
let's go.
I know Elizabeth Eschel, who is really interested in this topic. Megakaryocytes are on the top of the scattergram. When you look at the high side scatter, the megakaryocytes are reaching near the end of the side scatter on the top of the scattergram. And then I can only gauge this population and look at the large DNA content. And I was able then to take these very nice pictures of the megakaryocytes when I was looking at the marrow.
Thank you, Jordi. Our next question. Can the flow cytometer be used for environmental samples?
Yeah, in fact, we are looking, I have a lot of experiments looking at seawater. It can be easily, we can easily run the seawater on the CytPix and look at the presence of different particles, not only microalgae. I'm really interested in the presence of microplastics in seawater, but also in fresh water. And I'm also able to detect small bits that are used in cosmetics preparations. And this is a very nice topic, of course. As I showed in my presentation, it can be applied to the detection of microalgae, but we are always we are also looking at the presence of bacteria and other microorganisms especially in water. But using filtered air, you are also able to detect these particles after preparing a suspension and look at the presence of these very small microorganisms on the CytPix.
Thank you, Jordi. Now, there's a lot of movement towards imaging flow cytometry lately. Do you think reviewers will be looking for imaging data to confirm flow cytometry data? And have you had any reviewers ask for images?
Yeah, absolutely. It is. I fully agree that there is a growing interest in this field. And of course, the reviewers will ask about this specific information. In fact, I'm preparing a manuscript. I have not the feedback from the journal, but at the end they will ask about this specific information because it's crucial for the identification of rare cells, rare populations. In addition, we are confirming that in the singlet regions, we can identify doublets, and we can identify doublets and aggregates in the after discriminating single cells. And I think that this will change the conventional flow cytometry ideas that were shared during many years, and this imaging technology will help to boost the flow cytometry field in the near future.
And Jordi, has the integration of imaging changed how you prep samples for your flow cytometry experiments?
Sorry, can you repeat your question?
I sure can. Has the integration of imaging changed how you prep samples for your flow cytometry experiments?
Yeah. Sorry for missing the topic of this question, because this is a very relevant and important topic, because sample preparation will affect different methods to prepare the samples, will affect the morphology. And this is a very important topic, because by using different fixative solutions, we will have different morphology. In general, the things will be similar, but at the end, different fixatives and different solutions will affect the morphology of the cells. That's the main reason, because we always try to avoid the use of fixative agents, but it's not always possible. But at the end, we prefer the no lyse, no wash methods, because they will preserve better the morphology of the cells that we are running on the CytPix.
Very good. And Jordi, let's wrap with this final question. Does the addition of imaging make running your experiments slower or more complicated?
We are increasing the data that we are collecting. This will affect the conventional files that we are obtaining on the flow cytometer. And this means that we will need more time to process this information. The acquisition on the flow cytometer is the same that conventional flow cytometry. This will depend on the number of the images that we are collecting, and this will affect not the time on when we are running the cells on the flow cytometer. This will affect only the size or the weight of the file that we are collecting on the flow cytometer. Then we need to learn more about how to combine the classical morphology, the classical flow cytometry analysis with this new morphology approach to increase the quality of the measurements that we are obtaining on the flow cytometer.
Jordi, thank you for this excellent Q&A session. Do you have any final comments you'd like to leave our audience with?
Yeah, I think that we are facing a new challenge in the flow cytometry field, and we need to be open-minded to this new kind of methods. I think that maybe, if not all the flow cytometers in the future will be equipped with this or different imaging technologies, and even the cell sorters will use all this approach. And I think that during the first days I was using the CytPix, I was really fascinated with the images I was collecting. I never used fluorescent labels, because I was trying to understand and to learn about the morphology of these cells using this brightfield imaging camera, which is a little bit different in comparison with the conventional optical microscopy images that we are obtaining in the hematology lab or using optical microscopy. After learning more and better understanding the images I was collecting, I started to perform the combination of the classical immunophenotyping with the morphology approach, and now I'm very satisfied with the results I'm obtaining, even because we are confirming the true nature, as I was saying in my talk, to be sure that we are identifying rare malignant cells, such as in the case of multiple myeloma at low concentration.
Well, I want to extend a huge thank you to you, Jordi, for your time today and your important research. I also want to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Now, before we go to our audience, I want to thank you for joining us today and for your interesting questions. The questions we did not get to, and any of those questions that come in during the on-demand period, they will be addressed via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and Labroots will send you out an email to let you know when it's available for replay. Now, we encourage you to share that with your colleagues who may have missed today's live event. Thank you so much for joining us and spending your morning with us. Have a great day, everyone.
Thank you.
Until next
You're
time,
welcome.
bye-bye.
Bye-bye.
Thank you, Jordi.
Bye. Thank you.